First speaker, Debraj Ghose from Harvard. He's going to talk about immune cells. Thanks.
Hey, thanks for having me. I'm Debraj, and I'm going to talk about how immune tissue behaves as this shape-shifting active material, and I would really like to be able to understand it and control it. Before I begin, I'd just like to start with acknowledgements. My postdoc advisor, Donald Ingber, for the support and giving me the freedom to pursue this project and many useful discussions. Tom Ferrante for a lot of the microscopy that I'm going to show you. And that's about 1/3 of the Ingber lab and many folks at the Harvard Medical School that told me when I was, that corrected me when I said stupid things about immunology. So very broadly, I'm interested in biological self-organization and how these systems can compute. And this can happen across a range of scales. Previously, I worked on how self-organizing Rho GTPases and their effectors can generate these assemblages that search for each other, which is how yeast cells can mate. At the level of cells, cells can self-organize into tissues and organs, and organisms can self-organize into these beautiful collectives. And of course, collectives can self-organize into environments. So my long-term goal is to develop a biophysical understanding for these types of collective behaviors and then be able to control it. And ideally, I'd like to do this in a context where we can come up with biomedical solutions. And I think the immune system is a framework that could allow me to do this. For instance, within lymphoid organs like tonsils, spleen, or lymph nodes, you have the emergence of structures such as germinal centers. These beautiful structures consist of things like B cells, T cells, follicular dendritic cells, macrophages, fibroblasts, and many other cell types, which ultimately allow B cells to evolve and produce specific antibodies. This doesn't just happen in lymphoid organs. It can also happen near tumors. And these are known as ectopic or tertiary lymphoid structures, and they're often associated with better patient outcomes. You can think about it as the immune system setting up a military outpost closer to where you have your intruders. It can also be pathological, such as in patients with rheumatoid arthritis. They will display, again, beautiful structures, but extremely harmful in the synovium. And even outside the body, when you challenge cells from a tonsil or other immune cells with antigen, they will start to self-organize into these structures as they're generating an adaptive immune response. And what I want you to take away from this is that immune cells and other cells associated with the immune system have this intrinsic capacity to self-organize. Now, these structures are often fluid-like, and they can rearrange themselves, which is critical to immune function. And this kind of liquid-like physics has been appreciated in things like intracellular condensates, and also in early development, where many of the same principles of phase separation apply that allow the generation of patterns that initially start out being liquid-like, but over time, as the tissues mature, they become solid-like. What I think makes the immune system so special is a lot of immune tissue maintains this fluidity. And the word I like to use is protean, which means it can shapeshift. So my interest is in being able to harness this ability of immune tissue to shapeshift. And on one hand, we could control natural processes, such as generate or control germinal centers or tertiary lymphoid structures. And on the other hand, this is a kind of material that we could potentially use to build machines with novel functionality. But to get there, we need to understand immune tissue as this active material. So I decided to focus on the germinal center as a framework, though this could apply to many other types of immune structures as well. Within the germinal center, B cells that have diverse receptors compete for antigen, which is a piece of the intruder. The one that's best at binding to the antigen internalizes it, chops it up, and presents it to follicular helper T cells. These T cells give B cell signals that allow them to differentiate into plasma or memory cells, and many of the other B cells then proliferate while mutating to introduce more diversity into the receptors. These newly mutated B cells will then compete for antigen again. So this entire cycle represents Darwinian selection of B cells, which is how you end up with specific antibodies against intruders. I decided to capture this as a multi-component Kawasaki-Ising model. So what you're seeing here are a bunch of B cells that are yellow, which represents a low affinity state. And I can put down the rules that I told you, and these rules are sufficient to lead to the evolution of B cells. What's important in this model is this term here, that's effective temperature, how much the cells are moving around. And as you change effective temperature, you'll see that there is an optima in terms of what the affinity maturation can get to. And the intuition behind this is that if effective temperature is low, then the tissue is behaving more like a solid, so you can't have the rearrangements necessary for affinity maturation. If it's too high, the tissue could break apart. So my question now is, can we control this effective temperature? Are there ways in which this active material could control its state? So a place to look for it is where B cell states are affected. And in this cycle, germinal center reaction that I just showed you, T cells provide a bunch of critical signals to B cells. So if we look at the same signals that affect the state of B cells themselves at a single cell level, could it also affect the material properties of immune tissue? It has been known for a long time that when you stimulate B cells with these germinal center factors, such as CD40 ligand, they'll start to form these aggregates. And for brevity, I call them B lymphocytes organized as multicellular blobs, or BLOMs for short. And so I start by thinking about what are the material properties of these BLOMs. I can track these BLOMs over time. That is, I just get outlines. And what you're seeing here is a BLOM that changes, that evolves from 0 to 35 minutes. And you might appreciate that at 0 minutes, it's not very circular, but by around half an hour, it starts to become more and more circular. And this applies to BLOMs of different sizes. So this is characteristic of liquid droplets rounding as a consequence of surface tension. Now, the other thing that liquid droplets do is that they can mix. So if you, once again, go back to this Kawasaki-Ising representation of cells, if I have two BLOMs of different colors next to each other, what I can show is that as you increase this effective temperature, you go from being a solid to being more liquid-like, to then being dispersed like a gas. Similar if you change the adhesion. And intuitively, you can think of a bunch of particles, right, that are subjecting each other to forces, and when you have more motility, it behaves in a more liquid-like fashion. So experimentally, I can do this to see where BLOMs exist, and I can color some of these BLOMs white, leave the rest to be black, and ask what happens to these boundaries. So when I run this experiment, you see that it homogenizes, and so BLOMs behave like these liquid droplets. So stimulated B cells are sufficient to produce these types of multicellular condensates. But can we control the material properties of these BLOMs? Going back to our model, intuitively, if you have a droplet and you increase the effective temperature, you'll notice that the circularity of a droplet will start to drop. There are many ways to analyze this, but this is one of the intuitive ways to think about what happens to geometry as you increase effective temperature. So in the germinal center reaction, I can try to look at other factors that affect B cell state and ask what happens when I expose BLOMs to these factors. So what I observe is that in the presence of factors, cytokines such as IL-21, there's an increase in effective temperature. So the bottom row are BLOMs that have been exposed to IL-21. And I hope you can kind of appreciate that these have more raggedy edges. They're less circular. And this is clearer when I just quantify them. So in the presence of IL-21, which are the two violin plots on the far right for these two different donors, you see a reduction in circularity. So my point is that tissue fluidity seems to influence immune function. And my claim is that cytokines can tune this tissue fluidity. And this gives us a new axis to think of how we could potentially control immune function. Now, we can also start having a little bit of fun with the BLOMs and think about what we could do outside the body with them. One of the things the BLOMs do is if I track these using neural networks and just look at what these three guys do, they move around on the tissue culture plate quite a bit. And a lot of this movement is often quite persistent. And my thought is that these bloms, in order to move, they have to interact with a rigid substrate. And the reasoning is that there's something known as the scallop theorem that says that symmetric movements by an entity in a low Reynolds number regime will not really allow you to swim or get anywhere. You need these coordinated movements. This also applies to random movements. So in this case, bloms which are slightly buoyant can't be swimming. They have to interact with some kind of substrate. And you do kind of see that when you label the actin and look at cells near the bottom of bloms, you see them transiently interacting with the surface. If I now build a computational model around this that takes forces into account, what we observe is that over shorter timescales, you can get these rotational effects. So these things are capable of mixing over longer timescales, but over short timescales, you can transmit mechanical information from the substrate to the rest of the structure. So do bloms do this? Well, I can label the nuclei of these bloms and image them over time. And what I observe, and these guys, they're kind of walking around, they look kind of drunk. And what I observe is they start out kind of random, but then they'll roll. They do this rolling thing over a time scale of tens of minutes. Now, among the stimulations that I used, IL-4 was one that didn't lead to like a drastic increase in effective temperature. So I wondered if I could try to program this rolling behavior using IL-4. So here are 3 videos where I have bloms with IL-4 and without IL-4. And hopefully you can appreciate that on the far left with IL-4, the bloms tend to move faster. They also meet up more often, which causes them to get larger. It's a little clearer with the quantification, where on the top graph, we have mean velocity over time of these bloms. And as they're higher, they're more likely to meet each other, which leads to larger bloms. And so in conclusion, I guess the takeaway that is that I think cytokines can control immune tissue fluidity, and this could be a way to control immune function. And I think that these whole body dynamics of bloms could be programmable. And with that, I'm happy to take questions. Thanks.
Okay, we have time for, I think 2 questions. Raise your hand. We have mics.
Oh, yeah.
Beautiful presentation, thank you. My question is about the in vivo relevance of your findings.
Yeah, so one of the reasons I'm using like this blom system is because I started thinking about, okay, how do I think of active material properties in the context of something like the germinal center? And eventually I want to get to a point where I can study it in an actual germinal center. But right now, theoretically, I'm able to show that, look, changing the fluid properties can affect affinity maturation. So, but how that does it in like an in vivo context, that's not clear yet. Those are the kinds of experiments that I would want to do in the future. Yeah. Yeah.
Oh, hello, I have a question. I think I missed the point that does addition of the lymphoid cytokines increase or decrease the fluidity of these bloms, and is the changes in the blom motility dependent on an increase or decrease in the fluidity of the system itself?
Right, so these cytokines, they seem to increase the fluidity. IL-21 seems to have a much more drastic increase, which changes the blom's geometry. In terms of motility, it's the ones where the shape doesn't change so much, where you can maintain this rolling behavior. It appears something like IL-4 can increase motility. And this happens as a consequence of both slight increase in internal effective temperature, as well as the cells just being more active on the edges. Yeah.
One more question. I think we have time.
Nice to talk to Debraj. Do you observe Ostwald ripening between these multicellular condensates?
In this case, I don't think this is Ostwald ripening. It's more active. There are other cell types, like if I look at jerkats, there they seem to show things more similar to Ostwald ripening, which, I mean, we've done before. But yeah.
Cool. Let's thank him again.
Cool. Thanks so much.